So thank you very much to ILSI for inviting me to speak, and I'm really excited to be here today. Hopefully what I have to say will be of value to you. So my disclosures, my sources of funding are the Canadian Institute of Health Research and the Seeding Food Innovation Grant Program funded by George Weston and Loblaws. So I'll be talking about proteins and a little bit of proteins and protein quality, the methods to measure protein quality, factors that affect protein quality, studies in adult males that I've been doing, and some consideration for children and older adults, and some conclusions and recommendations. I'm not going to try to persuade you with regard to whether or not plant-based diets are the way to go. I think it has been, I think there's consensus that plant-based diets would be more environmentally friendly, and so there is some general agreement on that. And so I'm not here to convince, rather to present some of the research that I've been doing on plant-based diets and how we might make those diets nutritionally adequate. So protein is an indispensable or an essential macronutrient, and it's made up of 20 amino acids. They include the indispensable or indispensable amino acids. How do I do? Okay. Indispensable, conditionally indispensable, and dispensable amino acids for the purposes of protein quality and dietary concerns. The indispensable amino acids are the ones of main concern. Now, current protein recommendations, according to the Institute of Medicine and the FAO, WHO, is based on high-quality protein. High-quality protein is basically animal proteins. These proteins contain all of the indispensable or the essential amino acids in the correct proportion needed for protein synthesis and bodily function. They are also highly digestible when it compared with plant proteins. And so the key point here is to recognize that providing energy needs are met, the ingestion of adequate dietary protein is the most critical nutritional factor to support growth and maintenance of lean body mass in individuals of all ages, and that the most important aspect of proteins from a nutritional perspective is their amino acid composition, particularly their indispensable amino acid composition. Now, current amino acid and protein requirement really illustrate the importance of protein quality, especially as it relates to plant-based diets. Current recommendation for lysine, which is an important indispensable amino acid because it's limiting in plant-based diets, in the 1985 FAO-WHO report was 12 milligrams per kilo. In the last report, the 2007 report, the requirement estimate for lysine has been more than doubled, 30 milligrams per kilo. However, the protein requirement recommendation has virtually stayed the same. 0.6 moved to 0.66, 10% grams per kilo. This illustrates how important it is that diets are protein dense. And so how do you meet your requirement for lysine in plant-based diets that are limiting in lysine if the protein requirement is that low? Important to understand is that the World Food Program sets quotas for food based on requirement recommendation. So this is a very important point. So the question then is, can the human dietary protein and amino acid requirement be met by plant proteins? And we've understood for generations that a plant-based diet is the mainstay of the dietary intake of people in developing nations. In developed nations, we do eat a lot more animal foods. We understand now that for sustainable diets, we need to move toward a plant-based diet. And so that is important from a sustainable perspective, and from a nutritional perspective, we do have data that plant-based diets are beneficial to health as well. Plant foods, however, cereal grains in particular, as I've said, are limiting in the essential amino acids lysine and tryptophan, whereas pulses, which are the chief protein sources in plant diets, are limiting in the sulfur amino acid, particularly methionine. And so, in order to understand to what extent plant-based diets are health-promoting from a nutritional perspective and whether or not they can meet the dietary recommendations for protein, we need to understand the quality, the protein quality of these diets. And so dietary protein quality depends on the amino acid content as well as the bioavailability of these amino acids. And bioavailability includes digestibility as well as utilization of the amino acids for protein synthesis. And so the study of protein quality by looking at amino acid bioavailability bridges the gap between protein requirement and the amount of food that is required to meet those requirements. And this is particularly important for diets that are plant-based. So just some definitions. Digestibility refers to the proportion of the amino acids or a nutrient that is transferred across the intestinal tract after digestion. Bioavailability includes the proportion of the dietary amino acids that are absorbed but are available for protein synthesis, because some amino acids might be absorbed, particularly lysine, but not available for protein synthesis. And so our group have coined the term metabolic availability to combine both digestibility and bioavailability. Now, there are many factors in plant foods that are not in animal foods that affect bioavailability of amino acids and hence protein quality. Trypsin inhibitors in foods like soybean and red kidney beans, these can considerably affect amino acid digestibility and absorption of many of the amino acids in soy proteins. These can be inactivated by heating up to a certain point. Beyond that, too much heat can actually decrease protein quality. Tannins are present in many cereal grains and also in some pulses as well. They are not affected by heat, but some of them are affected by soaking, alkali treatment, and they can also decrease amino acid digestibility and protein quality. Phytates, very common in plant foods, very common in cereals, also common in some of the pulses. They are relatively heat stable as well, but they can be to maybe 30% of the phytates in some of these foods can be removed by extrusion at different temperature and moisture. And so all of these factors can decrease amino acid bioavailability, but also micronutrient bioavailabilities, particularly phytates, calcium and zinc and iron, these magnesium, these are affected by some of these anti-nutritional factors that are present in foods. Maillard reaction products are formed by the reaction of lysine with reducing sugars as a result of heat and processing. And so the lysine could be damaged and absorbed from the intestinal tract, but not available for protein synthesis. And so this can have effects on protein quality as well. And so what are the implications then for humans consuming a predominantly plant-based or solely a plant-based diet when amino acids are not available in foods? Then more food is required to meet the protein and amino acid requirement. And so we need to correct for the bioavailability of the amino acid to meet the recommendations. And this is very important, especially for plant foods that are high in fiber and high in these anti-nutritional factors. So again, in order to understand to what extent these anti-nutritional factors impact the quality of the protein, we need to study the protein quality of these foods. And so there are many methods in the literature, but in terms of the more acceptable, widely used methods, PR, protein efficiency ratio, has been used for many years and still used in Canada and in the United States for infant formula, as I understand. And so this is a rat growth assay. There's lots of assumptions, and the data isn't always readily transferable to humans. And so PDCAS, protein digestible corrected amino acid score, many of you will know about. It's been used for almost 30 years, and it is it was the accepted method for determining protein quality of foods for human consumption, approved by the FAO WHO. It is a chemical score, and it's based on the amino acid composition in the proteins relative to its requirement. It's corrected for fecal digestibility, however, and the fecal digestibility are usually obtained from rat assays. They truncate the scores to 100%, and it doesn't account for anti-nutritional factors that are present in the food. So there have been many limitations of using the PDCAS, which was recognized by the FAO, WHO. And so in 2011, they convened a meeting, the FAO WHO convened an expert meeting in Auckland, New Zealand, to look at dietary protein quality evaluation in human nutrition. And so one of the main decisions from that meeting was that amino acid will be used as individual nutrients for protein quality evaluation. And so why is amino acid availability in diets important? Well, only a portion of the amino acids in foods are digested and absorbed and used for protein synthesis. And availability of amino acids different between foods. So we understand for animal foods, it's much higher compared to plant foods. And that amino acid digestibility is different than protein digestibility when it's assessed using fecal nitrogen. There is data which suggests that amino acid digestibility for most of the amino acids are much lower than when digestibility of a protein is measured using fecal nitrogen. And so amino acid digestibility, we know, has been used by animal scientists for many years, but largely ignored in humans. And so the recommendation was that digestible indispensable amino acid score will replace the PDCAS. And so this is definitely an improvement over the PDCAS. It uses ileal digestibility of amino acids instead of fecal nitrogen. However, this method is very, very invasive, and so it's unlikely to take off in humans for measuring protein quality of foods for human consumption, because it requires nasal or endoileal tube placement, and that is in Canada for sure, no one is going to sign up to do such an experiment. It has been done by the French group and others, the New Zealand group, Moan and Hodgkinson. They're using the pig as a model and patients with ileostomies. They're trying to validate that work. But the committee agreed that based on the available data in the literature, there was not enough data to actually put the DIAS at work. And so in 2014, there was another meeting convened in Bangalore, India, to look at research approaches and methods for evaluating the protein quality of human foods. And so the goals of that meeting was to identify one or more experimental protocols to enable the development of more robust data set for true ileal amino acid digestibility in human foods, and to recommend best practice for the assays of true ileal digestibility, but to look at direct measures of amino acid bioavailability involving stable isotopes. And so this was one of the publications, the chief publication from that meeting, apart from the booklet, of course. And this was looking at the key characteristics of an ideal method to determine bioavailability of amino acids in proteins in human foods. And so some of the key characteristics of an ideal method is that it should be a method that directly measures protein quality in humans and that captures the effect of processing and cooking, that it should be minimally invasive, cost-effective, and validated in humans. And so our group applied the indicator amino acid oxidation method to the study of protein quality by looking at the bioavailability, which we termed metabolic availability of limiting amino acids in foods for human nutrition. And so some of you will be familiar with the IAO method. It has been used, it is a stable isotope-based method. It has been used for many years, over 25 years now, to determine all of the indispensable amino acid requirement in adults anyways. It has been applied to determine indispensable amino acid requirement in neonates, as well as in children, as well as in vulnerable populations like children with PKU and people with MSUD, maple syrup urine disease, as well as in individuals with liver and kidney disease. And so it's a minimally invasive method. It's based on the partitioning of an indispensable amino acid, and that's usually, we use that amino acid phenylalanine between oxidation and protein synthesis. It's based on the premise that when one indispensable amino acid is limiting in the diet for protein synthesis, then all other amino acids are oxidized to the level of the amino acid that is limiting. And so when, if you look at this diagram, to determine the protein requirement, when an amino acid is limiting and the diet can be increased from deficient to excess, we measure the response of phenylalanine by using a tracer, 13C phenylalanine. And as we increase the intake of the test amino acid, there is a decrease in the oxidation of the indicator amino acid phenylalanine until the break point, which is the estimated average requirement. And then we can determine the 95% confidence interval by looking at the variance around the mean. Now, this we have applied to study the protein quality of foods by looking at the indicator response to feeding graded intakes of phenylalanine in the form of free amino acids, as well as looking at comparing the response to of the phenylalanine oxidation to the intake of the test food. Now, this method is minimally invasive, as I've said. Each level of intake is usually tested over 3 days, 2 days of adaptation, and 1 indicator amino acid oxidation study day. Subjects are exposed to a deficient or excess intake for 8 to 9 hours, and the isotope is given orally. We sample breath for the purposes of these studies instead of blood, and every study day has about 4 hours of adaptation and 4 hours of isotope infusion. Now, the same individuals can be studied over several levels of intake because of the shorter adaptation period. It's non-invasive, and so it can be used in vulnerable populations like children and the elderly, and as well as individuals with disease and chronic conditions. And so the study day protocol is we do a pre-study, the study protocol is a 3-day protocol. There is a pre-study assessment, weights and heights and body composition and calorie requirement. And then there is a 2-day adaptation to the diet, and on the 3rd day, the subjects are given hourly meals, and the isotope is given at the 5th meal hourly. Hourly doses of the isotope, and breaths are sampled before the isotope starts and about 2.5 hours after, when we get to an isotopic steady state. Carbon dioxide production is measured, and this particular study design is around a 9-hour study day. And so this just shows you graphically how we determine the availability of the amino acids in the food. So we generate a reference curve by looking at the indicator amino acid oxidation response to amino acid intake fed as free amino acids, in which we vary the intake of the amino acid, the test amino acid that we are looking at, lysine or methionine, and then we feed the protein-bound, the food with the protein-bound amino acid, and we look at the ratio of the indicator amino acid oxidation response of the amino acid in the test food relative to the reference protein. And so the calculation of availability, we look at the oxidation. As I said, oxidation changes per gram of protein-bound amino acid divided by the oxidation changes per gram of free amino acid, which is equal to the proportion of the amino acid in the food which is available for protein synthesis. And so this method was validated in the in pigs by Moen et al. In 2005. And so this is the reference curve generated by using various intake of lysine in response to free amino acid, and this was generated by feeding the same intakes of lysine from peas, and then heated peas. The peas was heated, which suggests production of Maillard products, and as the peas was heated, you can see the metabolic availability or availability of lysine decreased. It was 89% in the peas, 55% in the heated peas. And when the heated peas was fed with a proportion of lysine HCl, you got a change in the oxidation to that of the reference protein. So these studies demonstrated that the method is sensitive to changes in heating and processing of the foods. And so we applied this method in humans to determine the lysine availability in white rice. And so this was published in a table form because one of the reviewers had asked us to change the graph to a table, and so we did. And so we assume that the free amino acids are used 100%, and so the lysine availability in the cooked white rice was 97%. Then we browned the rice in an oven, and we again studied the response to the oxidative response to changes in the lysine intake, and so the availability dropped down to 70%. This again demonstrated that in humans, the method is also sensitive to changes in heating. We also published on the metabolic availability of lysine and tryptophan in maize, in white African cornmeal. We imported this cornmeal from South Africa, and the metabolic availability or bioavailability of lysine was 71%, the availability of tryptophan 80%. So what this tells us is 80% of the tryptophan in the cornmeal after cooking, as you would a pap, a porridge, as they do in South Africa, only 80%, well, 80% is available, 80% of the tryptophan is available, only 71% of the lysine is available. We also use this method. This is recently shown at ASN conference in Boston. This is one of my students' master's project looking at the availability of lysine in millet, pearl millet. This is a common millet consumed in India. And again, this was interesting data. We did not expect that the metabolic availability would be so high, but it was 85%. Now, this is the reference curve formulated from the free amino acid mixture, and this is the millet curve. Now, the difference with the study is that we introduced the effect of complementation. These studies are a little bit difficult to do because we use a repeated measures design, and so we enroll healthy adult men, and we study them at each of the 4 levels of intake of lysine from free amino acids, and then we study the lysine from the millet in this case, and in the same subjects, and so this is a repeated measures design, and so in order not to lose these subjects, sometimes we have to make a guesstimate as to what the availability would be, and so we estimated that the availability would be about 75%, and this was using digestibility from pig data that we had for millet, and it was done using millet that was ground up in pigs. And so we estimated 75%. It actually was 85%. So using that, we did a complementation of the millet with lentils to see if we can decrease the oxidation of the indicator amino acid, which is the inverse of protein synthesis. So a decrease in oxidation suggests that more is taken up for protein synthesis. And we can see that the oxidation curve decreased further than the reference curve, and this was 108% of the reference curve. And so this does make sense because we estimated 75%, it's actually 85%, and so the oxidation, this shows that we've augmented the lysine intake to be above that which was provided in the reference diets. And this was a similar study, again, another one of my students looking at the availability of lysine in Indian wheat. And so we ordered the wheat from India, and we looked at the availability of the lysine. Again, this is the reference curve. This is the wheat curve, and the wheat was served as baked bread. And again, we complemented with lentils. We estimated 75% again, and then the metabolic availability was 89%, and then we fed the lentils. We were able to augment the lysine supply so that the slope of the line, the regression line, showed a 120% that of the reference slope. And so we also did this study with chickpeas alone or combined with rice. And the metabolic availability of methionine, this was looking at methionine, the limiting amino acid in chickpeas, was 68%. When we combined with rice, we achieved the same level of oxidation as that of the reference curve, because here we estimated an availability of 65% in this rice. And so when we look at this is the amino acid composition on the milligram per gram, or if you think about it as the amino acid score, this is the amino acid composition of lysine and methionine and leucine. Have added leucine there in egg protein, millet, and those that we've done. This is the scoring pattern as per the FAO-WHO, which suggests that for a food to be adequate in lysine, it should provide 45 milligrams per gram of lysine and 22 milligrams per gram of methionine. And here we see that each of the foods that we have done corrected for the availability is lower than the 45 for lysine and lower than the 22 for the methionine. And so the implications for children, we have not done studies in children. However, in children, the protein to energy ratio is quite low for these foods, and so we would have to do some studies to see what these are. Now, this is a study looking at lysine requirement of children in India who were partly malnourished, but clinically they seemed okay, but they had intestinal parasites. So before treatment and after treatment for intestinal parasite, the lysine requirement was determined, and it was 20% higher before treatment and after treatment. So certainly we understand why children living in challenging environments, it's difficult to meet their requirement, because it does seem that for lysine anyways, and eating a plant-based diet, the lysine requirement is higher. And so so I'll just skip this slide, sorry. For adults, there's lots of data which shows, and this is from the Adventist study, which shows that a vegetarian diet is very beneficial, and there's lower mortality, all-cause mortality in adults eating a vegetarian diet. The implications for older adults, we know from a lot of the data that's available in the literature from Stu Phillips' lab and Patton Jones that older adults have a higher requirement for protein to stimulate the same level of protein synthesis compared to younger adults. And per meal, older adults seem to require about 30 grams compared to 20 grams. Also see from rat data that digestibility is lower in older than younger animals. And so we're not sure, but we do need to do these studies. We know that the population is aging, and there are more people over 60 than ever before in modern history. And so protein is very important. And so to summarize, using the IAO method, we have determined for some foods the availability in adults. We need to do, it's a very viable method to use for vulnerable population. It can be used to test the effect of protein complementation of plant foods, and there's implications for transitioning to a plant-based diet. Takeaway, protein quality of a wide range of foods need to be determined across different stage of the life cycle, special consideration for infants and growing children. We also need to be cognizant of micronutrient deficiencies and special consideration for adults. Just a recommendation based on the data that we have derived so far, combining grain with chickpeas in a 3-to-1 to 2-to-1 ratio should be able to meet the recommendation for lysine and protein in adults. Just to acknowledge Penchars and Ball, these are my mentors, and my collaborator, Rajveli Lango, and my lab technician, and the students who've done the work.
Thank you for your presentation. I'm curious about the, I think it was a few slides before the end, on the children that were malnourished and had the infection. So we know that children who are malnourished and have an immune system that is compromised or digestive system that is compromised have a higher requirement on lysine. Is there a consensus now of how much that requirement is increased by?
For that particular study, the requirement was increased by 20%, but they had no clinical symptoms, so we don't have data on children who are symptomatic. It's possible that it could be higher, we don't know. But for these particular children, they had intestinal parasites, and the requirement for lysine was 20% higher before they were.
So we can't say that 20%, if we increase by 20% the lysine for
we don't know that for sure.
Yeah, okay.
For this particular study which I didn't speak about, by Shabini Ghosh, she looked at data, balance sheet data from 118 countries, and she looked at the percentage risk of protein inadequacy. And you can see, before correcting for protein quality, the risk was about 12%. After correcting for protein quality, closer to 35%. After correcting for intestinal enteric dysfunction and risk of an infection, then it went up to almost 50%.
Thank you.
So lysine plus total protein, that's probably all factored in that study.
Thank you, very intriguing. Christy Reimers, I'm with Conagra Brands. I'm interested in your slides on the complementary proteins where you were feeding lentils and millet or lentils and wheat. And if I'm understanding it, it showed the bioavailability was higher when those complementary proteins were fed together. And it makes me think that for years our recommendation to vegetarian or vegan eaters is you don't have to worry about eating those foods at the same time. As long as you get them over the course of the day, you'll do fine. Do we need to change that recommendation, especially for people who are eating vegan diets who may not be having any excess protein in their diets?
Yeah, I think more and more the data is showing, and especially for older adults, that the timing of the intake is even more important than the total intake across the day. And so eating these foods at the same time is what's going to stimulate the muscle protein synthesis in all of the studies that's been done. And so for younger adults, 20 grams of protein. For older adults, 30 grams per meal is what stimulates the protein synthesis. And so there's studies looking at older adults eating where the protein per meal is given, whether 50, 60, 80% of the protein versus just eating the normal pattern of eating where the protein's smaller amount, 11 grams for breakfast, 15 grams for lunch, and a whopping 30, 40 grams for dinner. Protein synthesis is much better if you eat that where you're giving for each of the meals 30 grams of protein compared to the way we do eat in North America right now.
Can I ask one more, since there's not a line? The other thing that was interesting to me were the anti-nutrients. That's becoming a big buzzword in popular nutrition blogs, a way of keeping people away from oftentimes whole grains, lentils, the fear of the phytic acid, the tannins, et cetera. How much of an issue do you think those components are relative just to, getting adequate protein, adequate diets? Is that really something that should be emphasized, or is it being overemphasized?
So these are naturally occurring substances. They're important for the plant. Some of them are what they call phytochemicals. So in that context, they're important for the plants. That's why we need to understand if they're present to a large extent in foods, and they're present to different extent in different cultivars of the same food, how much of the amino acid or the nutrients are available, then we can adjust. Beyond that, we don't need to make much more of those anti-nutritional factors.
Thank you.
All right. Any other questions?
So I'm interested in your thoughts about potential fortification of grain foods with lysine.
So that's one strategy that has been tried in the past. It's been tried in India. It's been tried in many developing countries. They've also, there's been genetic engineering of various grains like sorghum and maize in particular. That's been done. The thing about the fortification programs is that a lot of them, the results, the data didn't show an improvement in growth in children because they didn't know what the starting point was. So even if that's going to be one of the strategies that's used in developing nations, let's say, we still need to understand how much of the amino acid is available before we know how much to add. And that's one of the problems because in a lot of the studies, not enough lysine was added to these foods. Of whether it's wheat or sorghum, they didn't add enough, and so they didn't see. And for sorghum and some of these other grains, different cultivars, the anti-nutritional factors are far more. Like we used in this study, pearl millet, let's say. We have a study going on now with sorghum. And for certain millet, the digestibility could be as low as 60 or 50%. And if you don't know the availability, you just don't know how much to add when you're fortifying.
All right, last question, Moha?
Another question, but just to comment. Alongside with the components that are in the food that might decrease the availability, probably you've seen it. There was an article 3 weeks ago in Science showing that actually the exposition to CO2 on the multiple cultivars of rice decreased the content of protein. So we should really be testing the amount of proteins very regularly because CO2 does decrease the amount of proteins in our food.
Yeah, you're right. That data is out there, and people are doing. And for all of our foods, before we start the studies, we do send them for analysis as well.